Very nice turnout tonight. I appreciate it. Keep sharing this with your family and friends and neighbors. And there ain't no word of mouth like word of mouth. That's a tautology. Tonight it's my pleasure to welcome Cameron Currie back to Wednesday Nite The Lab. He's a professor in bacteriology. He was born in Edmonton, Alberta. And he went to high school in Edmonton. And he stayed in Edmonton to go to the University of Alberta, where he got his bachelor's degree in biological sciences and his master's degree in entomology. Then he went over to Ontario to get his PhD at the University of Toronto, which is where my father-in-law went for undergrad. And then he got his first faculty position at the University of Kansas in Lawrence, in Missouri.
I did not tell Tom that.
Then in 2004, he came here to UW-Madison. Tonight, he's going to talk with us about pretty intriguing stuff, the secret lives of leafcutter ants. Would you please join me in welcoming Cameron Currie back to Wednesday Nite The Lab.
Thanks, Tom. This is really great to be here. It's my actual third time at Wednesday in the lab. I'm really proud of that. So it's great to be back. One thing I forgot was how funny Tom is. He sets a high bar for kind of humor. I'm going to try to stay away from some of the slapstick stuff. But yeah, so it's great. So has anyone been at any of my previous Wednesday Nite in the Lab? Put up your hand. Tom? Okay. Did anyone, we tweeted out from the Currie Lab, anyone on Twitter following the Currie Lab Twitter account? The Currie Lab people and friends are all friends. So that's terrible because we sent out homework last night to watch the last Wednesday Nite in the Lab online, which is where I'm going to start right at the end of that. No, I'm just teasing. Yeah, it's great to be here. You know, when Tom asked me to come back, I was excited and had some things in mind and picked the date well in advance. I didn't realize how big a week it was going to be. You know, some things happened this week. Like on Sunday, it was Super Bowl, right? And so we had that excitement. On Monday, it was the Iowa caucuses, counting day one. Tuesday was Iowa caucus day 2. And today, other than Wednesday at the lab, is what? Iowa caucus? People obviously don't follow us on our ant Twitter. It's the big ant day, right? That's the big thing that happened today, right? That's all we're caring about today is the ant day. So part of my talk is to really kind of introduce you to the secret lives of leaf-cutting ant. Some of it's not going to be so secret. Some of it will be more secret stuff, and really kind of give you some of the amazing biology of leaf-cutting ants. And so. Last time I was here, I talked a lot about kind of the role of symbiosis and microbes and symbiosis in shaping the diversity of life. And I asked people how many people have read this book and know who E.O. Wilson is. How many people in this room have read this book or know of this book? Good. So I thought I'd start with a quote from Bert Hawdobler and E.O. Wilson from a different book of theirs that I think really kind of illustrates why leaf-cutting ants, are so amazing, or kind of points this out. If a congress of naturalists were to gather to choose the 7 wonders of the animal world, they would be compelled to include the bizarre, mighty civilization of the Attine leafcutters. And it's, I think, hopefully my major job in the first half of my talk is, for those of you who are not convinced the leaf-cutting ants are a wonder of the world. Hopefully by the end of the first half of my talk, you're convinced. So I'm going to focus, as I said, first half about a lot about cool stuff about leaf-cutting ants, the basic biology. And part of the reason for this is we have this display of our live leaf-cutting ant nest in the Microbial Science. How many people have actually seen this display? Good. How many of you want to see this or have not seen it? Good, good. So at the end of today, after my talk, I've volunteered to give a personal tour of the ant display. So if you have time after and you want to walk over with us, you can get a personal tour. We are super excited because we have new live webcams that are on our ant colony that Caitlin worked very hard to produce, and here's the links here. And so please check out our new webcams that are live 24/7 and really a great kind of outreach part of this approach. So the first half is going to be amazing stuff about leaf-cutting ants, and then in the last half I'm going to talk about a little bit, small snippets of what we do on leaf-cutting ants. And since I was here last, we've published a lot of work on leaf-cutting ants, and there's lots of really great work going on in the lab by people in this audience that I can't speak about. I can't fit all in. So I'm going to pick a few snippets to talk about the complexity of leaf-cutting ants. And I'm going to end with an effort to kind of link the basic science that we do on leaf-cutting ants to some applied outcomes to really kind of make the case for why basic science is so important. And I do a lot, in my lab does a lot of outreach, including maintaining this exhibit on the ants to kind of educate the public about the importance of basic science and the fascinating natural world. And a part of that is to make the case of, basic science has applied outcomes. And so hopefully I'll be able to have time to end with kind of the potential clinical implications of what we're doing. So I've got some videos at the start here. I think videos show leaf-cutting ants the best. Special thanks to Adrian Smith. This is his Twitter handle. Please follow him on Twitter. He's got some amazing stuff online showing leaf-cutting ants. And so I'm going to run a few of his videos here. So this, how are the lights? Down a bit? I'm going to show that one more time. One of the cool things you'll notice is the mandibles of this ant are actually serrated like a steak knife to make them more efficient cutting through the leaves. And so that's really incredible. If you want to see the other side of the process, this is the worker with her serrated mandibles slicing through that leaf fragment. And she stridulates her abdomen to signal to other workers to come and forage on that leaf fragment and to signal where she is, and does this incredible behavior. Obviously leaf-cutting ants cutting leaves. One ant, one fragment of leaf, super cool. But really what's incredible is seeing the action over time. This is a sped up cutting of a leaf fragment by leaf-cutting ants, making rapid work of that leaf fragment. Pretty impressive. Who's seen leaf cutting up close? Good. Good. So then they cut the fragments and they hoist them over their heads and they carry them back to the nest and make along these worn trails. And so here are workers running back to the nest with leaf fragments, and you can make out workers running to the plant leaf to cut more leaves to carry it back. An impressive display. And just one more video to start off, some more later. But this is one of my favorite. Just the activity of the ants. There are going to be tens of thousands of ants cutting leaf fragments, carrying them through the forest. An incredible, incredible sight. Who's seen leaf-cutting ants actually in the tropics in the field? Good. Good. This is my favorite part. It's all the ants. I've seen columns of leaf-cutting ants 12 wide with ants carrying leaves. Just an incredibly impressive feat. So they cut leaves, they make trails, and they carry them back to their nest. And really, it boggles the mind, the scale of leaf-cutting ants. This is a savanna in Paraguay from the upper atmosphere. This was taken up by a former grad student in my lab named Jared Scott using Google Earth. So this is taken from 1.85 kilometers on Google Earth. These little dots are active leaf-cutting ant mounds that you can see from the upper atmosphere. Amazing, amazing feat. So these are cows on the surface of the soil of the savanna. Really an amazing superorganism. The estimate is there's 40 tons of soil with taking billions of ant loads to remove from inside the ground to make this leaf-cutting ant nest. Super impressive, right? Well, what's the most mind-blowing part is what occurs underground. A group poured 10 tons of cement over 3 days into a nest and excavated the colony. And this is the savanna. These are people excavating the soil to expose this nest. For those of you who are worried that this was an abandoned colony, they didn't pour it on live ants. Some people get all excited about that.
Cameron, can you turn the lights back up for the folks at home, please?
Sure. So it should be all on, Tom? How's that? Okay. These researchers actually estimate the amount of work one individual ant colony engaged in to make a nest is the equivalent of, on a human scale, of making a Great Wall of China. Just an incredible, incredible superorganism. Very impressive. Leaf-cutting ants play important roles in natural ecosystems. They stimulate new plant growth. They help break down vegetation. They excavate soil and stimulate root growth and help with filtering of nutrients deeper in the soil. This is one of my favorite photos. This is a Mark Moffett photo that was in National Geographic in the 90s. This is a savanna. These are active mounds. And you can see actually where there's abandoned nests, there's enough nutrients to support tree growth. You know, really illustrating the important role in nutrient cycling. It's not my phone. Okay, what powers the system is, of course, mutualism, cooperation, a beneficial association that is one of the textbook classic examples of mutualism in nature. The ants actually don't eat the leaf fragments. They use it to grow fungus for food. And so they're farmers farming fungus for food. This is a leaf-cutting ant worker on a little fragment of leaf material. You can see the green here. This is the filamentous growth of their fungal mutualus growing on the surface of this. The ants produce specialized feeding structures, or the fungus produces specialized feeding structures for the ants that we call gongolidia that are thought to be nutrient-rich and kind of optimized to support the growth of the colony. So the benefit of the ants from this association is nutrition. They get the nutrition from the fungus, and in exchange, the ants provide the benefit of the fungus in dispersal. I'll show you that a little bit later. I hope I convince you they go to great lengths to get substrate for growth of the fungus, and they engage extensively in protection. Linnaeus, the godfather of taxonomy in the 1700s, described the genus Atta of leaf-cutting ants. I think it was in the 1750s. Thomas Belt in 1874 discovered the mutualism between the ants and the cultivar. And so we've known about leaf-cutting ants for a very long time. They've fascinated people, indigenous people and Europeans throughout European history. There's really fascinating reports in the literature about Portuguese settlers into the New World, writing reports back to Europe about how the leaf-cutting ants were really affecting the colonization and the ability to colonize the New World. And so leaf-cutting ants have had a big impact on kind of aspects of human biology, human colonization from the perspective of their foraging activity on plants. And so we've known about them for a very long time, and they've fascinated humans for a long time. I'm going to go through some other fascinating facts about leaf-cutting ants. This is the royalty part of leaf-cutting ants. This is the queen ant. She's incredibly robust, amazing individual worker. She can live 10 to 15 years in her lifespan, lay 200 million eggs, an incredible feat. Stores 300 million or more sperm in her spermatheca for those 10 to 15 years and continually use it. Amazing feat to be able to do that. And oversee colonies composed of millions of workers of ants. So those big colonies, the big nest will only have one queen. So that's the majestic royalty. The less majestic, sorry guys, is the male. This is a male leaf-cutting ant. Notice the scrawny head, wimpy legs. The wings are actually poorly attached. Males, leaf-cutting ants, live a few months. They mate with a few females and then they die. The ant colony only puts the minimal amount of energy into production of males to survive the nuptial flight and mating. And the queen is incredibly robust and strong. So if you have any questions, please put up your hand. Does anyone have any questions at this point? Sure.
How long does the average worker live?
Great question. The average worker, we think, lives around 6 months. So the average worker lives much longer than the males. All the workers are female, and the queens are female and the males. Yes, at the back of the room.
Is there more than one queen per colony?
There's only one queen per colony that's actually functioning as an egg-laying queen. You may have sister queens that get mixed up and don't go on their nuptial flight. They'll dealate and they'll become a worker, but they won't lay eggs. Okay, so amazing royalty. There's also complex division of labor in terms of what the workers do in the colony. And the first way the division of labor works is an age-based division of labor. And so the youngest workers do the safest tasks, and they tend the fungus garden, the brood, and they do all the kind of the careful cleaning of the fungus garden. They don't leave the nest. They don't actually leave the fungus garden, and they do these tasks. When they get a little bit older, they go to tending the fungus garden, to leaving the nest and foraging. And then they get even older, they start taking out the trash. They also, when they get older, they engage in wars with other ants. They get even older, they take out the trash. And then finally, they tend the dump, they tend the trash pile, the oldest workers, and they eventually die. So in ant societies, in leaf-cutting ant societies, the youngest workers do the safest jobs, and the oldest workers do the most dangerous jobs. And that is, obviously predicted to be, optimization of energetically. So you want your youngest workers at the longest lifespan still to serve the needs of the colony to do the safest jobs. And so you have this age-based division of labor that's really fascinating. Leaf-cutting ants also have a size-based division of labor. This is one of my favorite photos. This is a soldier leaf-cutting ant with her huge head that's enlarged to maintain very large mandibular muscles to make her strong at cutting. And then this is what we call a minimal worker, whose size is really thought to be ideal for tending the fungus garden, moving around the nest. There's actually 7 or 8 distinct size ranges of workers, and these different workers do different jobs within the colony. So a little bit about the colony founding and colony growth. The way leaf-cutting ants reproduce is they produce new queens and males that are winged, who go on their nuptial flight. So this represents a mature leaf-cutting ant nest. And these here, if you can see them, are actually flying ants. They're actually super impressive to see in the field. The queens, the new queens, take a little pellet of fungus from her parent nest to start her fungus garden, and the males, because they don't help found colonies, do not bring the fungus. Then the queen will dig a little chamber underground, and she'll spit up a little fragment of fungus garden, and she'll start out on her own. So she leaves a nest of millions of workers and starts a colony on her own. And then slowly she'll lay eggs, she'll have a few workers. Maybe about 8 to 10 months later, she'll start to have a significant workforce of dozens of workers. And then you get into exponential growth, and 5 years later, you'll reach a colony this size. So this production of reproductives is large in the mature colonies with a lot of mortality, as I'll talk about in a minute, of new queens on the nuptial flight.
What's the doubling time for colony size?
Double size of a, so it increases over time. It's actually something I am not sure has been ever worked out. One of the challenges with this kind of work is all this stuff is occurring underground, and to dig it up, you disrupt it. And then in the lab, it's so artificial. So we do know, as I've marked some nests and followed them, they go from a queen to having workers in 3 or 4 months to having, several thousand within a year to 100,000 in 2 years. And so there's a definite exponential growth. Don?
How frequently does this the swarming behavior occur?
So that's a great question. This actually is synchronized seasonally, and it happens once a year or once or twice a year at the beginning of the rainy season. So colonies produce reproductives coming up into the beginning of the rainy season. So the tropics don't have a winter, summer, spring, fall like we do. They have a wet season, dry season, and so the reproduction occurs at the beginning of the wet season. So they produce the new queens and they go nuptial flight, and it's really incredible because colonies of different species of Atta in the same populations will have different times of night where they'll actually have their nuptial flight, and so they're out of sync in terms of their time. So they have their own window of about 2 hours where they'll release their reproductives. Great question. Does that make sense? Okay, so incredible biology. Individual queen within years reaching colonies of massive size. Leaf-cutting ants produce a lot of refuse or dump, so they actually have specialized workers, as I mentioned, that take out the trash. And what you can see under here are little fragments of partially decomposed leaf fragments that have been removed from the bottom of the garden, carried out by workers, and dumped. They'll run along a vine or a trunk, and they'll actually dump them on a specialized mound. I've seen refuse mounds that are this high where workers will run up the side of a tree and go up top and drop off their refuse. They have elaborate waste management practices, which is thought to kind of keep the microbial contaminants that accumulate in the dump from getting back in the nest. And so there's a really kind of hygienic part of the life cycle of these ants that are maintaining the refuse in kind of an isolated location from the garden. Really amazing aspect of this system too. So leaf-cutting ants have uninvited guests. This is one of our favorite. This is actually a cockroach. Most people aren't big fans of cockroach guests, but these are actually kind of cute little cockroaches as cockroaches go. So this is a little cockroach that occurs in the fungus garden. These cockroaches actually are only known from leaf-cutting ant nests. They will actually, the thought is they actually will ride on new queens to found new colonies. I've seen these cockroaches follow trails of ants when they move to new nests along the trail to go to their new colony, and so they're really an amazing aspect of the system. The other favorite uninvited guest is discovery of a former PhD student in my lab named Heidi Horn. It's one of my favorite stories. Heidi went down to Costa Rica with us, and before we left, she's like, "I'm really excited about digging up leaf-cutting ants, but I'm a little scared of snakes." And I'm like, "Oh, well, we barely see snakes. You know, it's not going to be a problem. You know, and you know, we have boots and stuff." And I showed her how to dig a leaf-cutting ant nest. And at this point, I dug maybe 2,000 nests myself. And I showed her how to dig the nest, into the nest, and I said, "Okay, go to it, Heidi." She started digging, and I let her kind of do her thing, and I wandered off a bit, and I hear the scream, and I run over, and she'd found a snake inside the garden. And so she was freaked out, and I was so excited because I've dug 2,000 nests. I've never seen a snake. Heidi, on her first colony, finds a snake. And so it really is incredible. Both the cockroach and the snake are going to have chemical signatures that mimic the ants to kind of camouflage themselves in the nest, and really amazing biology. The major predators of leaf-cutting ants are army ants. It's really incredible to watch an army ant raid a leaf-cutting ant nest. There'll just be hundreds of army ants running down the nest, coming out with their brood. They're not stealing the fungus, they're stealing the brood of the ants. I've never seen a giant armadillo myself, but I've seen the activity of a giant armadillo digging in a nest, and they have a major impact. The queens on the nuptial flight are prey to a lot of different animals. Bats at night eat them. Small vertebrates will eat leaf-cutting ants on their nuptial flight. People also eat leaf-cutting ant queens. How many people have eaten insects here? Has anyone eaten a leaf-cutting ant nest queen before? Just me? There's some new people in the lab who are here who are going to the field, and this is a good chance to tell them that this is a ritual of the lab to eat these queens. Actually, indigenous people in South America will actually eat these new queens, and you think, well, one queen in this big nest. Well, they don't dig up the nest to get the one queen. They actually will catch the queens coming out of the nest when they're on their nuptial flight. And these indigenous people actually know when these queens are going to come. They've learned about the cycles of the ants, and they'll be able to watch the colony and changes in the colony to know when to watch for collecting these queens to eat, and so it's incredible. One of our favorite pests is actually a parasitoid wasp. It's a forward fly. Forward flies will lay their eggs in the head of other and bodies of other insects. The larva will consume the tissue of the insect, and then it'll emerge as a new fly. These are actually really small. This is the fly here, their wings. This is a worker of, I think it's an Acromyrmex leaf-cutting ant being attacked by this forward fly. Really incredible. If you're a fan of the movie Aliens, that's kind of what's natural world is going on here. Really amazing biology. And part of how leaf-cutting ants are thought to combat forward flies is actually having what we call hitchhikers. And so this is a larger worker carrying a leaf fragment, and this is a smaller worker riding on that leaf fragment, and this is the hitchhiker. And the thought is, and there's some evidence to support this, that when the forward lands on the leaf, because it needs a perch to lay its egg in the head of the worker, these hitchhikers will scare the forward off to protect the worker from the forward fly. And so just really incredible behavior that's evolved in the system. Any questions about that? Cool. Okay, so the diversity of leaf-cutting ants. I've been talking largely about the genus Atta, which has 15 species. It's probably an underestimate with cryptic species. but currently 15 described species. They form these massive colonies with this large worker polymorphism, as I showed you. The sister genus is called Acromyrmex with 24 species. They have large colonies as well, but not millions of workers, but hundreds of thousands of workers, and they don't have the same extent of the worker polymorphism. This is my favorite leaf-cutting ant, in part because of these two white patches that I'll be talking quite extensively about later in my talk. Okay, so still amazing biology, but I'm actually getting into the non-entomology part of the talk, more the microbial stuff, which is actually what my lab does. I'm a professor in the Department of Bacteriology. As Tom said, I did my master's in entomology, but we mostly work on microbes now. Microbes associated with hosts, largely leaf-cutting ants, but other systems. And the first microbe I want to introduce you to is the fungi in the genus Escovopsis. And Escovopsis is a member of the Is anyone here a mycologist? One or two few mycologists? People in my lab are, good job, guys. The fungus the ants grow is a member of the Basidiomycota, and the pathogen Escovopsis is a member of the Ascomycota. And Escovopsis this is what it looks like in culture. This is its micromorphology. So it's these vesicles with phyllides and conidia produced on it. This is the anamorphic state of the fungus. Escovopsis is only known from ant-fungus gardens. We've shown that it parasitizes the fungus the ants grow, so it gets its food directly from the ant cultivar. It's horizontally transmitted between nests. That means it's moved between nests by some vector that we don't know, or through the air. We've shown it's not vertically transmitted with the queens, and as I'll show you in a second, it's a very virulent parasite of the system. And so one of the amazing things about the ant system is they maintain these gardens in monoculture on these large scales, and obviously when humans maintain large farms and monocultures, we have problems with disease and weeds and pests. And so the ants are very similar in that regard. They have crop pathogens, other fungi in the genus Escovopsis. Escovopsis can be devastating. This is a healthy nest of a fungus garden, a healthy nest of a fungus-growing ant. This is one that I found in the field that had been completely overgrown by the pathogen Escovopsis. And so this is the partially degraded substrate the ants were growing their fungus on, and this is all the mycelium or the filamentous growth of the fungus that had completely overgrown the nest. Every once in a while when I go on a trip, I'll come back and someone who's looking after our ants in the lab will be all upset because we had a whole nest overgrown by Escovopsis in the lab, and they'll be really upset, and I'll be really excited because the pathogen won. So the fungus consumes the fungus, as I said, and so this is the growth of Escovopsis, and this actually is the fungus the ants culture for food. So this is the cultivar, and so the Escovopsis will actually grow towards the cultivar in culture, and when it comes in contact, it will actually stimulate more growth and explode and consume the fungus. In our hands and the work we've done so far, we've found no evidence that the cultivar is able to protect itself from its typical pathogen Escovopsis that infects its nest. And so this suggests that the cultivar is being provided protection by the ants and other mechanisms. And we do a lot of work on these mechanisms. The first mechanism I want to talk about is what we call fungus grooming. And so this is a leaf-cutting ant, minimal worker with her antennae detecting the presence of experimentally infected, sprayed-in spores of Escovopsis. She's actually running her mouth part along the mycelium of the fungal cultivar and collecting the spores that we infected the nest, and accumulating these inside a structure we call the infrabuccal pocket inside her head. And so one way I like to think about it is imagine the spaghetti is attached to your plate, and you got spaghetti sauce, and the spaghetti sauce is the pathogen, and the ants just basically running the spaghetti through their mouth and lapping up the spores of the pathogen. And this behavior we call fungus grooming, and is a really common behavior in the presence of infection. As I mentioned, they'll accumulate these spores in the structure called, we call infrabuccal pocket. This is a sagittal section, the head of an ant. So this is the head. This red here represents accumulation of spores of the pathogen that have been lapped up by a worker, made it into a pellet. And what's really incredible, and I expect everyone to say this with me the second time, is they make infrabuccal pellet piles. Infrabuccal pellet piles. So they pile up these piles of was that a Tom joke? That makes me a Tom joke. Sorry. The regulars get that. These are actually accumulations of these pellets that they'll actually maintain in a specific location in the nest. And we've published a few papers on this, and we actually haven't worked on this in years, and it's an area that would be really interesting to get back to. We have some evidence that these piles will sterilize the spores of the pathogen. What we mostly work on in defense is actually a second mutualism in the system, bacterial symbionts that produce antibiotics. This is a leaf cutting ant in the genus Acknowledgments, and this ant is not brown or red like other ants completely, but actually is covered in this whitish coating. This whitish coating is a filamentous bacterium in the genus Pseudonocardias. I'll talk about it in a bit. I often ask this question. I won't, because there's a bunch of people in my lab will know the answer. But what filamentous bacteria like Pseudonocardia are really well known for is the production of antimicrobials. Most of the antibiotics we use in the clinic are actually derived from this same group of bacteria that are on the ants. And so this is what the bacteria looks like in culture. As I mentioned, it's genus Pseudonocardia in the phylum Actinobacteria, which are well known for producing antibiotics. So if you actually culture the bacteria from the ant in nutrient media, so this is a SEM close-up of the two white patches of the bacteria on the ants. If you culture it in artificial media and you challenge with the pathogen in the fungus garden, you get these potent zones of inhibition. And what this indicates is that the bacterium is secreting some chemical into the media, and when the fungus comes in contact, it's suppressed. And so it says there's some antimicrobial being secreted, so antimicrobe, and some susceptibility in the fungus. And so this fungus is not resistant to what's being produced. And that's a really important point of this. It's not just about what the bacteria is producing, but also the sensitivity of the fungus. And so I have a depiction here of what this assay looks like, and this is actually what we call a zone of inhibition. Here's the bacteria from the ant, here's the pathogen, and the larger this zone of inhibition, the more potent, we assume, the least suggests more potent antimicrobials. And so this is the growth of the fungus along the edges of the media, but it doesn't grow into the plate where the antimicrobials are. Does that make sense? So we do a lot of work on this, and I'm going to come back to this when I talk about what my lab is up to. So one of the other cool aspects of the association with the actinobacteria is the bacteria is actually integrated into the social structure of the ants. And so this depicts this. This is a pupa. So this is, like if you think about caterpillars, they go and butterflies. They go from eggs to larvae that feed on the plant, and then they make a pupa, and then they eclose to being a butterfly, right? Ants are like that. They're whole metabolous insects like butterflies, and so they have a pupal stage. They go from egg to larvae to pupal stage. The pupae, when they eclose to adult, do not have the bacteria that produce the antibiotics. But we've shown they get inoculated by other workers with the bacteria, and in 14 days they have a heavy coating of the bacteria all over their bodies. And so we actually like to call these workers sheep. It's like they have this sheep's coating. And what's really incredible is these workers that are sheep are embedded in the fungus garden where the pathogen is. And then when they get older and they go to foraging, remember there's this age-based division of labor, they actually lose the bacteria. So the bacteria is integrated in its role in terms of tending the fungus garden into the social structure of leaf-cutting ants, and, which is really quite amazing. Okay, so I think this is my second last slide on kind of the fascination of leaf-cutting ants. And I really need to point out that I've been talking about leaf-cutting ants, but leaf-cutting ants are part of a group of even more ants that grow fungus for food. And so there are two genera out of a tribe of ants that are New World tribe of ants that tend fungus for food. And they form, as you saw, these incredible massive colonies that were super impressive. But most of the diversity of fungus-growing ants is cryptic. And so this is our leaf-cutting ant nest with their two patches of antibiotic-producing bacteria. This is actually the size of about a quarter. This is in dirt here, and inside here is the whole colony of this Cyphomyrmex ant. This whole colony is the size of one of the larger marbles. So this ant is incredibly cryptic compared to the big, massive colonies that I showed you, and really impressive. These are really wonderful to collect. Compared to digging up a big ant nest that has soldiers that can bite through leather shoes, that can draw blood, that take hours and hours in the heat. Sorry guys, I'm making you think about all the fun you're going to have in the field. These ants, you just take a little tweezer and you roll the whole nest into your container. It takes 5 seconds. Beautiful, clean collection. Wonderful to work on. The only problem is they're very hard to find. Other fungus-growing ants actually can form nests inside bamboo. So bamboo, this is the fungus garden. This colony would be, about that large. And another group of fungus-growing ants actually will grow their fungus not in the mycelial state, but in the yeast form. And really incredible biology of growing yeast gardens. And so this is the filamentous growth of the fungus, and what you can see is this kind of wet-looking growth of the yeast form of fungus. And so there's a huge amount of diversity of leaf-cutting ants. Most of it's cryptic, and it's co-evolved over tens of millions of years. And so this is a representation of the evolution of fungus-growing ants from Ted Schultz and Sean Brady, adapted from their 2008 paper. And this is a depiction of the diversity of fungus-growing ants. And what we have colored on here is the different agricultural associations of fungus-growing ants. The main point here is the ants are really diverse, and as they have diversified over time, the fungus they grow have diversified as well. And so there's ancient coevolution between the ants and their cultivar that has occurred over 55 to 60 million years, all throughout the New World tropics. This is a representation of the fact that we think that fungus-growing ants evolved in the Amazon basin in Brazil. The distribution of the ants is represented by the gray. There's actually fungus-growing ants that get into the pine barrens in New Jersey. There's leaf-cutting ants in some of the southern eastern states, Texas, Louisiana. Has anyone seen leaf-cutting ants in the U.S.? Not just in my lab? Yeah. In Texas? Yeah, collecting leaf-cutting ants in the U.S. is a little bit nicer for the permit side of things, right? Importing ants from tropical countries, we have to get special permission. But the challenge is leaf-cutting ants in the drier habitats of places like Texas are really deep, and so it's a huge trade-off to dig those nests. And so we prefer to go to Costa Rica and Panama. Okay, so how am I doing for time? Getting low on time, so I'm going to have to go a little faster. I hope I've convinced you the fascination of leaf-cutting ants, and studying them and understanding their biology is really worthwhile from a basic science perspective, and hopefully you're excited about coming and seeing the leaf-cutting ants. In my remaining time, and I'm going to run out of time, I'm going to address a little bit of the applied aspects of what we can learn from leaf-cutting ants and why studying, as I indicated at the beginning, how basic science can inform on relevant stuff like in biomedical. And obviously, this relates to antimicrobials and discovery of antibiotics. These ants have antibiotics, have bacteria that produce antibiotics. This could serve as a source of new antibiotics. And so I'm going to get into that. I'm going to skip this slide. I'm going to talk really quickly about the evidence we have that the ants have been using antibiotic-producing bacteria for millions of years. And so this is a leaf-cutting ant in the genus Akromyrmex. This, again, is a sheep that develops in 14 days. These workers actually have specialized structures on their body called tubercles that are connected to glands that feed the growth of the bacteria. So major modifications for maintaining the bacteria. These get even more elaborate. This is another fungus-growing ant in the genus Myrmex. This is the bacteria here. I bleached off these bacteria and discovered they have very large crypts or cavities for maintaining the bacteria. We sectioned through this. This is the cuticle of the ant. The purple is the bacteria. These are the glands. There's channels. These ants have very elaborate morphological modifications for maintaining the bacteria, with these indicating that this is a really important part of the system and evolved over time. These structures get even more elaborate. Here's the spot that I just showed you. This ant is completely covered in white dots, these openings that are full of bacteria. And if you sagittal section through the Myrmex ant, as we did, this ant is actually completely covered in crypts full of antibiotic-producing bacteria. You can make an argument that this is like a walking pharmaceutical factory. Major modifications for maintaining the bacteria. We recently, led by a former postdoc, Hanji Lee in the lab, published on the evolution of these structures. I wish I had time to go into detail, but there's no way you could see this anyway. But I have to show this figure because it took Hanji huge amounts of time to produce it, and it's absolutely beautiful. But Hanji's major discovery was he discovered there's 3 independent evolutionary events of these elaborate structures on these ants for maintaining the bacteria, and 4 losses. So there's a really incredible evolutionary history associated with these bacteria on the ants. And we published on the presence of the bacteria on ants in Dominican amber. Dominican amber is about 15 to 20 million years old. The ants have been associating with these antibiotic-producing bacteria for at least 20 million years, and based on our evolutionary work, probably from the origin of the association. They've also had there's a lot of variation in the chemistry that's produced. This is former work. This work was done by a former postdoc, Mikhail Polson. And Mikhail just basically challenged 12 different strains of Ascovopsis against 12 different strains of bacteria from the same population. And the major point here is red is strong inhibition, white is resistant. The major point here is it's not all red. In the same population, different strains of bacteria produce different antimicrobials. There's variation in the chemistry. And we've actually, with natural product chemists, have been characterizing this diversity. A lot of this work was done with John Clardy, who's at Harvard Medical School. More recently, we've been working with Tim Bugney at the pharmacy school here in Madison, and Monica Pupo, who's at the University of San Paulo in Ribeirão Preto, characterizing the antimicrobials that are being produced by the bacteria on the ants. And these are all new to science and of interest to as potential antimicrobial drug leads. One of the amazing things that a former postdoc in John Clardy's lab, in collaboration with us, discovered was that a nest of fungus-growing ants just kilometers away from another had a different variant of the same family of antimicrobial compounds, suggesting that the chemistry changes on the kilometer basis in the terms of the antimicrobials. Now, I'll remind you, I showed you leaf-cutting ants and fungus-growing ants go from southern Argentina up in the Pine Barrens. So chemical variation on this scale is incredible in terms of this, thinking about the full scale. We've been digging into the genomics of antimicrobial. Mark Chevret here is help with that. We have a paper just coming out on this. But the major point here is this is a genome tree. Each individual column is a different biostatic gene cluster that can produce an antimicrobial. And we can study the evolution of the genome as it relates to production of chemistry. And there's a predicted genomic diversity in the chemistry as well. I'm good for 5 more minutes. Can everyone hang in 5 more minutes? Tom, am I okay? 5 more minutes? Okay. He's on the mic. He told me I could go for 55 minutes. So, and all his jokes took some of my time at the beginning. So, sorry, Tom. So I'm going to end with a few slides here that relate to kind of what's developed from this, and you know. Ants using antibiotics, bacteria to produce antimicrobials to protect their fungus garden. Pretty cool. We thought this was really amazing when we discovered this is the first example of an insect using antibiotic-producing bacterium to treat disease. After we published on this, Martin Koltenposs group found a similar association in European bee wolves. Jared Scott, a former postdoc in my lab, found this in southern pine beetle. So it's not just present in leaf-cutting ants or fungus-growing ants. We were finding it in different insect systems. But the major user of these bacteria producing antibiotics are humans still, right? And so antibiotics are a really important part of our biomedical system. And you know, this is a slide from my colleague David Andes. I'll show you a picture of him in a minute. And to me, like as a non-biomedical person, this was a little mind-blowing to me. Like I knew about antibiotics being important for treating infectious disease, and you know, going from a society where doctors just diagnosed stuff and didn't have treatments to being able to treat things like infection was a major transition in the history of biomedical our biomedical history. But antibiotics are critical for organ transplants, complicated surgeries. You can't perform complicated surgeries without antibiotics or organ transplants. A lot of the compounds that are getting from antimicrobials are used in cancer chemotherapy, critical care medicine, care for premature babies. Our biomedical field depends on antibiotics, which is a bit of a concern, right? With emergent antibiotic resistance. Who here is concerned about antimicrobial resistance? Who's not concerned? When I teach, I like to ask the questions the way I want people to respond. You know, this is dated. This is I actually used this last time I gave Wednesday Night in the Lab, I think. So I think it's actually from 2010. I looked up the numbers. It's now 40,000+ people a year dying in the U.S. from antimicrobial resistance, and the estimate is 700,000 globally. One study recently published estimates that antimicrobial resistance deaths are going to exceed 10 million by 2050, which will make them more deadly than cancer, all cancers combined, by 2050. Really a major issue. If you have questions about this, come to me, or Mark up here knows a ton about this as well after. I just want to talk about how we're trying to contribute to this. This is Sir David Hopwood's in his Streptomyces in Nature Medicine. And this is from 1940 to 2000, the antimicrobials that have been developed and used clinically. And you can't probably see it, but most of these are in bold, and the bold antimicrobials are from soil Streptomyces. So Streptomyces in the soil. Streptomyces are part of the actinobacteria related to what's on the ant. And so most of the antibiotics we've traditionally gotten are from soil Streptomyces. Part of the reason we don't have new antimicrobials being developed is when you screen soil Streptomyces, you rediscover already known compounds. And so that's partly why pharmaceutical industries have abandoned this. Dick Boltz told us, he was involved in daptomycin discovery, that they screened 100,000 to maybe even as many as 10 million soil streptomyces to discover one antimicrobial drug. So this is a major thing. We have been discovering antimicrobials from insect systems at about 1 per 15 strains of strains from insects traditionally, and we leveraged that to generate a grant. Dave Andes and I led the first round to NIH to start the Wisconsin Antimicrobial Drug Discovery Research Center, which we just finished our first round of funding and initiated our second round. And so we're looking at symbiotic microbes, especially from insects, as new antimicrobial leads, including going down to testing in mice. And so this is a major undertaking in the lab. Tom, can I steal 2 more minutes? I got to do this because he's sitting in the second row, and she's sitting in the 5th row. Caitlin and Mark led our major undertaking of our work on this drug discovery center. This is a paper that we're particularly proud of. Huge amount of work, many figures. This is the figure that Mark made that is just beautiful and represents the extent of a lot of effort from Caitlin and undergrads in the lab of sampling. And what basically we have is circles representing different insect hosts that we sampled. And so you can see we have samples in Brazil, in the Amazon in Brazil, in Costa Rica, Puerto Rico, Florida, the Southeast, a lot around Wisconsin. Caitlin and a former person in the lab did a transect in Alaska. These are all host samples, and we sampled diverse insects from diverse biomes and studied these bacteria from these insects as drug sources. And Caitlin oversaw undergrads who ran 51,000 inhibition assays. And one of the things we found really exciting, so this is fungal inhibition by insect-associated strains, soil and plant. And what you can see is our insect-associated strains had more antimicrobial activity in culture than soil streptomyces. So not only are we getting new compounds when we dig into these strains, our inhibition assays suggest that they're more potent. And I encourage you to look at the paper. I'm not doing it near enough justice in terms of getting in the detail, but really exciting work that we've done. And so we've now, I think, are at 25 novel compounds from around 300 strains compared to the rediscovery rate of in the soil. And so really exciting source for antimicrobial discovery that we're doing. And this is David Andes, who's the head of this center, NIH center, and he actually takes antimicrobials and tries to cure human infection in mice in vivo, and is a world-leading authority on this. And Tom, I think you should have him out at Wednesday Night in the Lab. So I'm going to end with acknowledgments. You can see lots of Currie Lab members, current and past. You know, one of the great things about doing interdisciplinary science and working with really talented young researchers is, being able to see discovery through their eyes is really a wonderful thing. I talked about Mark, Hanji, and Caitlin's stuff in my talk today, and I have lots of collaborators that are not in here. I can only include the collaborators on stuff I talked about, and with that, I will take any questions. Thank you. Yes?
How does the ant biome system compare to the human biome system of our gut, sinus, and the number of different species and their ability to be pharmacists?
So the question is, how does the ant biome, meaning probably microbiome, right, in their gut compared to human?
Well, their environment.
And their environment. It's a really good question. I could have actually thrown some slides in there to actually show you data, but I had to cut some stuff and I already ran over time. So the first thing is the human. So we've been doing in our second round of research studying human microbiome as a source of antimicrobial, including the nasal, oral, skin, and we've done some work on gut. And it's really hard to fully assess at this point, but we've been surprised that the human source, some parts of the human source seem promising. What's important to know is that in our oral, nasal, and skin are members of the actinobacteria, which is what's on the ant. So our bodies in some locations are dominated by the same type of bacteria that are known to produce antimicrobials and are on the ant system. And so it's part of what we're doing. As I said, we've recently renewed the center, and one of our projects is focused on that, and I'm sure that one of the leads of that project will come and give a Wednesday night at the lab and tell you much more about it. But it's a great question. Yeah?
Yeah, I might have missed it, but did you talk about anything about something similar except in the New World, or is this same kind of ant systems exist elsewhere?
So the question is is there any, I only talked about the ants in the New World, is there any equivalent? And it's a great question. The fungus growing ants only occur in the New World. They originated after separation of South America from the other continents. After 70, 80 million years ago, they originated around 55, 60 million years ago. But in the Old World, there's fungus growing termites that occupy a similar niche. They're not taking fresh leaves, but they're using plant substrate to grow fungus for food and form massive colonies. If you've been to parts of Africa and Asia, you can see these very large termite mounds. Those are actually fungus-growing termites that are equivalent. There's other New World fungus-cultivating insects, and in fact, there's lots of kind of new systems that are being discovered that are being described like fungal cultivation, like ambrosia beetles, which have a global distribution. And they are super ecologically successful, but they don't form such large colonies as the fungus-growing termites and leaf-cutting ants. Great question. Yeah, Tom?
So I'm just wondering, when you found, I think, 4 losses of crypts,
yeah.
What kind of ecological shifts are associated with those losses? And the bacteria lost, too.
The bacteria, I think, in all 4 cases are lost. I know for sure in 3 of them, which is really fascinating. There's pathogens still. We know that in these systems, the garden pathogens there. So there has to be, some mechanism. In one of these shifts, we have some evidence that the cultivar might actually be protecting itself, but we have not been able to follow up on that. So that might be a role. Maybe behaviors taking a more role. There's some really interesting work by some members in this audience of whether other bacteria in the microbiome are taking that role. And so it's really an interesting question. But we only knew about 2 of those losses up to last year. Yeah, it's really interesting. Sorry.
Any of the local people in areas where leaf-cutting ants are a big part of the local environment make any use of the trash piles that the ants carry?
Yeah, so the question is, do any of the indigenous people in local areas where leaf-cutting ants occur make use of the trash pile? And it's a great question. Historically, I don't know, but I do know there's people who are now using that material to protect their plants because the ants really don't like the trash pile, and so people will use it as a skirting around there, as a barrier, chemical barrier, and so that's really interesting. I assume that there some nutrient fertilizer role could be used. It's but I'm not aware. But it's an interesting question, really interesting question. The dump is really an amazing niche. They build up in the dry season when there's no moisture for metabolism, and then they heat up when they're being in the rainy season. We've measured temperatures of 70 degrees Celsius inside these. So really amazing kind of bioreactors. So, Mark, you still want to ask?
Yeah. So you kind of alluded to a dual function of the infrabuccal pocket depending on what caste you're in. If you're a queen, you use it to take a fungus.
Yeah.
If you're a groomer, you use it to kind of weed out
Yeah.
invaders.
Yeah.
Is there anything known about the environment of the pocket in these different castes and how that might select for different microbes?
Yeah, so the question is about the infrabuccal pocket, and the queen uses it to carry the fungus on her nuptial flight, and the groomers use it to maintain the spores. And what do we know about the kind of environment in there? It's a great question, and the actual answer is we know almost nothing. The equivalent is like their oral cavity is this pocket. So is there microbes in there? And do they mediate the aspects of the biology? It's a great question. I think there's a real difference in size. Like when the queen carries this little pellet, her head is pretty big, and it's actually you can see the speck of fungus with the naked eye that she carries and rolls up. Really an amazing kind of feat. It may be that the microbiology of the pocket might help keep that garden clean on her nuptial flight, that might also sterilize the pathogen in the grooming, but we don't actually know very much about that at all. I think this is a great point to hit this point. Fungus growing ants, leaf cutting ants have been studied, and fascinated humans for a very long time. But and there's over 3,000 published papers in the system, one of the most well-studied systems. But we actually still are making major surprise findings that blow our minds, and that's one of the fun things about working on it. Like I told the story about Heidi Horton finding the snake. It just really is a fun system and a really deep system, and there's so many really cool questions still to work on. Kristen.
What's the diversity of the symbiotic cockroach that follows the ant trails to the colonies?
So the question is, what's the diversity of the symbiotic cockroach that follows ants? And unless it's been published recently and I have missed it, which is possible because it's, the pace of science right now is like drinking out of a fire hydrant. Morphologically they all look the same. I don't think there's been any description of the diversity. I've seen these cockroaches from across fungus growing ants, and they all look morphologically identical. Now I'll put on my microbiology hat and say, I'm not an expert on cockroach, and I'm sure a cockroach expert could look at that and go, oh yeah, there's really significant differences. But it's a really good question. And these cockroaches could be carrying around the pathogen that attacks the fungus garden. Is that what you're hinting at? Tom?
One other question. I'm just wondering whether anyone has studied how the tempo of fungal growth in the gardens depends on temperature, given that the
fungus
gardens are restricted to largely tropical or subtropical locales. Is it that lower temperatures would so reduce the growth rate that it's no longer a viable strategy at higher latitudes?
Yeah, so it's a great question. So leaf cutting ants are Tom's hinting at this. Leaf cutting ants get into Texas, but they haven't spread further north. There are fungus growing ants in New Jersey that are able to maintain their garden, but they're not leaf cutting ants. They actually will. I've never seen this, but I've been told about it. They'll actually take their fungus and break it apart in the winter, and they'll just hold them in their mandibles. So there's definitely a factor of needing to have a temperature to continually grow their fungus garden. There's some work out of the University of Texas, and that shows that the cultivar from more northern locations of leaf-cutting ants might be less or able to grow at lower temperatures. One of the aspects of the system that's really fascinating is the ants are pretty good at regulating temperature underground by opening and closing holes. So there's aspects of that that is that probably plays a role. But we know very little about this. There's some fungal cultivars the ants maintain that, in culture at least, will grow really fast, and others that grow really slow. And they're sister clades to each other, and the ants that maintain them each are cryptics, where we think there's speciation associated with switch in cultivar type. And at least in culture, these things grow super differently, and we know very little about that as well. Yeah.
You talked about army ants coming through.
Yeah.
How do the leafcutters respond to that? And do they survive? Do they just hunker down and let the brood go?
Yeah, so the question is, like, army ants attack, and how do leaf-cutting ants respond to that? I have some personal observation of this, and my personal observation of this is I'll make a joke, poorly. They handle it poorly. Army ants are incredible. I think it's a little bit of like they're raiding us, back away, let them take our brood, and we'll lay more eggs, right? That's kind of the impression I've had with a big army ant raid. Having said that, the soldiers will cover the queen, and they'll also take the brood, and they'll try to hide the brood. So a lot of that is occurring underground. It's hard to see, but when you watch, the army ants are coming up with the big larvae of the colonies, so they're like at a very high rate. So they're getting at some, they're getting the goods. But the soldiers will also kind of try to plug holes. But from my observation, it looks like the army ants are pretty good at raiding, and the leafcutters are probably tolerant, and they're like, oh, our queen can lay 150 million eggs, take 1,000 and be gone. Yeah.
Do ants encounter microbial resistance?
Do ants encounter microbial resistance? That's another great question. These are all great questions. The variation I showed indicates there's resistance in populations, and we've seen it in populations. And so the question is, well, how does the ants deal with resistance using this for tens of millions of years? And the snide answer to this is evolution is the result of that selection from resistance that generates the new chemistry, and so it's the it's the escalation of that is generating the new chemistry. And so in the clinic, we of course can't accept any resistant leaves to mortality. But having said that, there's really elaborate behavioral defenses, and the combination of these different defenses probably counters the selection of resistance in ways that we still don't understand. Last question, I think, and then come up to me after if you're, but yes.
Okay. The antibiotics derived from these ants, is it more broad spectrum use, or is there any use within precision targeted medicine?
So the question is, the antibiotics the ants produce are the broad spectrum versus targeted, and that's a great question, and the answer is a really hard one, and we think we're pretty proud of the number of new antimicrobials we've characterized, but it's still a very, very, very small proportion of what we know these things are capable of producing. So speaking to this is really hard because, you'd need to have more characterized. But if we do inhibition assays where we grow microbes against each other where there's production, the inhibition of the pathogen by the bacteria on the ants seems to be more targeted to the pathogen than general antifungals. Now, I'm talking about like I showed you some of the data from 55,000 assays. That is between insect-associated streptomyces and different human pathogens, but we've done tens of thousands of interaction assays between the bacteria in the ant and the pathogen in the fungus garden. And it's just a very complex thing that we still don't completely understand. It's actually, if you would have asked me that last time I gave Wednesday in the lab, I would have given you a really strong answer, like we knew this really well, and the more we learn, the more we realize we don't know. So, thank you.
Any other questions? Thank you very much. Next week, with Maria Rive, the folks that are organizing Darwin Days are going to have things called refreshments. Some of them will have to do with fermentation. We have to get a ferment. I don't not very good. And then Cameron will eventually, in the next few minutes, lead the group. Whoever would like to go over to microbial sciences and see the news.
Yeah, maybe we can ask who would like to go to see the colony. So there are people. Okay, great. So let's gather. Either wait for us at the back or come up to the front.